Hello friends, welcome to SRS by tutorial. I am Samiranjan. In this video, we will discuss about heteropolysaccharides. Polysaccharides are polymers of carbohydrate. According to their composition, they are classified into two types, homopolysaccharides and heteropolysaccharides. Storage polysaccharides like starch, dextran, inulin, glycogen, and structural polysaccharides like cellulose, chitin are coming under homopolysaccharide. Homopolysaccharide consists of only a single type of monosaccharide residue, whereas heteropolysaccharide consists of two or more different kind of monosaccharide. Glycosamine glycan is one of the common example of heteropolysaccharides. Glycosamine glycan. They are also named as mucopolysaccharide. They are long, unbranched heteropolysaccharide. They consist of repeating units of amino sugar and uronic acids. The amino sugar is either N-acetyl glucosamine or N-acetyl galactosamine. The uronic acid is usually D-glucouronic acid or L-diuronic acid. They are present in the matrix of cell wall and the ground substances of connective tissues in animals. Glycosamine glycan are classified into four types: hyaluronic acid, heparin, chondrotene, and keratin sulphate. Hyaluronic acid. It is a linear polymer of disaccharide units held by beta 1,4 glycosidic linkage. Each disaccharide unit composed of D-glucouronic acid and N-acetyl glucosamine joined by beta 1,3 glycosidic linkage. It contains alternating beta 1,3 and beta 1,4 glycosidic linkage. This figure shows disaccharide unit of hyaluronic acid. Hyaluronic acid occurs in the ground substances of connective tissue, synovial fluid, vitreous humor of eye, and it is the only glycosamine glycan which is non-sulfated. Functions of hyaluronic acid. It acts as lubricant and shock absorber in bone joints. It serves as cementing substances and contribute semi-permeability to membranes. It produces glycocalyx with glycosylated protein and help to adhere with each other in animal cells. Heparin. It is a sulfated glucosamine glycan. It consists of repeating units of D-diuronate 2-sulfate and N-sulfo-D-glucosamine 6-sulfate joined by alpha 1,4 glycosidic linkage. It occurs in blood and intracellular granules of mast cell that line arterial wall in lungs, liver, kidney, spleen, thymus and skin. This figure shows a disaccharide polymer unit of heparin. Functions of heparin. It acts as an anticoagulant in vivo and in vitro condition by inhibiting conversion of fibrinogen into fibrin. It also acts as coenzyme and increases activity of lipoprotein lipase activity, and hence it is called as clearing factor. Chondrotene. It is also called chondrotene sulphate. It is composed of repeating disaccharide unit of D-glucouronic acid and N-acetyl D-glucosamine 4-sulphate. They joined by beta 1,3 glycosidic linkage. It is present in the ground substance of connective tissue, widely distributed in cartilage, bone, tendons, cornea, heart valves, and skin. This figure shows disaccharide polymer unit of chondrotene. Functions of chondrotene. It is required for synthesis of osteocytes and cartilage. It has mild anticoagulant activities. It is a constituent of basement membrane and cell surface. Keratin sulphate. It is the only glycosamine glycan which does not contain uronic acid. It consists of repeating disaccharide unit of D-galactose and N-acetyl D-glucosamine 6 sulphate. They joined by beta-1,4 glycosidic linkage. It is found in the cornea, cartilage, and bone. This figure shows repeating disaccharide unit of keratin sulphate. Functions of keratin sulphate. In joint, they act as cushion to absorb mechanical shock. It participates in the development and in the glial scar formation.